friends, welcome back to my channel Only Bio. Continuing with the series of the NEET 2020 paper, the MCQs solved all in details with the explanation from 21 to 30. The first question you can say is an additional question, and the question is the product or products of reaction catalyzed by nitrogenase in the root nodules of leguminous plants is or are dash. A. Nitrate alone. B. Ammonia and oxygen. C. Ammonia and hydrogen. D. Ammonia alone. And the correct answer for this question is C. Ammonia and hydrogen. Root nodules are found on the roots of plants, primarily the leguminous plants, that form a symbiosis with nitrogen-fixing bacteria. Now, under nitrogen-limiting conditions, the capable plants they form a symbiotic relationship with the host-specific strain of bacteria called as rhizobia. And this process has evolved multiple times within the legumes as well as in other plants or legume crops, including beans, peas, and soybeans. Now, within legume root nodules, nitrogen gas from the atmosphere is first converted into ammonia, and this is then assimilated into amino acids, nucleotides, DNA, RNA, as well as other important energy molecules like ATP, vitamins, flavones, and hormones. And along with this, we find there's a release of hydrogen as well. The next question relates with the chapter Human Health and Diseases. It's a match the following having two columns. Column 1 A, B, C, D being typhoid, pneumonia, filariasis and malaria. And column 2 having the list of pathogens. First is Wuchereria, second is Plasmodium, third is Salmonella and D is Haemophilus. And the options given are, the correct answer is first, that is typhoid. You can relate it with third, that is salmonella. Pneumonia can be related with Haemophilus. Filariasis can be related matched up with Wuchereria, and malaria with Plasmodium. You all know typhoid is caused by Salmonella typhi, that is spread by drinking food or water contaminated with the feces of an infected person. The pneumonia is the infection wherein there is inflammation of the air sacs in one or both the lungs, which may fill with the fluid, that is pus, and the infection can be life-threatening to anyone. Viruses, bacteria and fungi can all cause pneumonia, but the most common being influenza and respiratory syncytial virus. A common cause of bacterial pneumonia is Streptococcus pneumoniae. Filariasis is a parasitic disease caused by an infection with roundworms. These are spread by blood-feeding insects such as the black flies and the mosquitoes. They belong to the group of diseases called as helminthiasis. And the lymphatic filariasis is caused by the forms Wuchereria bancrofti, Brugia malai, and Brugia timori. These worms occupy the lymphatic system, including the lymph nodes. In chronic cases, they may lead to a syndrome of elephantiasis. And malaria is also life-threatening diseases caused by the parasites transmitted to the people via the bites of infected female Anopheles mosquitoes. But however, it is preventable and curable as well. The next question relates with the chapter Evolution of Life, and the question is, from his experiments, S.L.Miller produced amino acids by mixing the following in a closed flask. And the options given are A. Methane, hydrogen, ammonia and water vapor at 800 degrees centigrade. B. Methane, hydrogen, ammonia and water vapor at 600 degrees centigrade. C. Methane, hydrogen, ammonia and water vapor at 600 degrees centigrade. D. Methane, hydrogen, ammonia and water vapor at 800 degrees centigrade. And the correct option is D. The Miller-Urey experiment, or just called as the Miller experiment, was a chemical experiment that simulated the conditions thought to be present in the early earth and tested the chemical origin of life under those conditions. The experiment was supported by Alexander Oparin's and J.B.S. Halden's hypothesis. We call it as Oparin and Halden's hypothesis that putative conditions on the primitive earth favored chemical reactions that synthesized more complex organic compounds from simple inorganic precursors. Considered to be classic experiment investigating abiogenesis, it was conducted in 1952 by S.L.Miller with assistance from Harold Urey. The next question relates with the chapter genes and it is, which of the following statements is correct? And the options are A. Adenine pairs with thymine through one hydrogen bond. B. Adenine pairs with thymine through three hydrogen bonds. C. Adenine does not pair with thymine. And D. Adenine pairs with thymine through two hydrogen bonds. And the correct option is D. Now, adenine is one of the two purine nucleobases, the other being guanine, used in forming nucleotides of the nucleic acids. In DNA, adenine binds to thymine via two hydrogen bonds to assist in stabilizing the nucleic acid structures. In RNA, which is used for protein synthesis, adenine binds to uracil. The next question relates with the chapter cell division and the question is, it's a match the following A, B, C, D being zygotine, packaging, diplotin, dikinesis, all of these being the stages of meiosis 1 and first terminalization, second chiasmata, third crossing over and fourth being synapsis. These are the options given and the correct option or the answer is 1. That is, zygotine can be related with synapsis, packaging can be related with crossing over, diplotin is chiasmata, and dikinesis relates with terminalization. Friends, look on the screen for understanding this. These are all the diagrammatic representation of the different stages of prophase 1. The second stage of the prophase of meiosis following leptotin is the one in which there is synapsis, that is pairing of homologous chromosomes, that is zygotine. Now the long Paquetin stage begins with the completion of synapsis and is associated with further thickening and shortening of chromosomes. During this stage, exchanges of chromosome material between maternal and paternal homologous chromosomes occurs by crossing over. At the points of crossing over, bridges, that is chiasmata, are seen in variable numbers. Diplotin is a late stage of prophase during meiosis in which the chromatic pairs of the tetrads begin to separate and chiasmata can be seen. The displacement of chiasmata is termed as terminalization, which is completed in the dikinesis stage. The next question relates with the chapter genes. It's a match the following again, wherein 1, 2, 3, 4 are polymerase, nucleases, exonucleases and ligases. On the other side, we have break the DNA into fragments, separate the two strands of DNA, make cuts at specific positions within DNA and join the two DNA molecules as the options on matches there. And the correct answer here is the D1, ligases joins the two DNA molecules. Friends, a polymerase, that's the option number 1. Remember, it's an enzyme that synthesizes long chains of polymers or nucleic acids. DNA polymerase and RNA polymerases are used to assemble DNA and RNA molecules respectively by copying a DNA template strand using base pairing interactions or RNA by half ladder replication. A nuclease, that's option number 2, it's an enzyme capable of cleaving the phosphodiester bonds between nucleotides of nucleic acids. Now, nucleases variously effect single and double-stranded breaks in their target molecules. In living organisms, they are essential machinery for many aspects of DNA repair. The option C or 3 is exonucleases, that are enzymes which work by cleaving the nucleotides one at a time from the end, that is exo, of a polynucleotide chain. a hydrolyzing reaction that breaks the phosphodiester bonds at either the 3' or the 5' end that occurs in the the exonuclease activity. And the option D or fourth are ligases, which are enzymes capable of catalyzing the reaction of joining two large molecules by establishing a new chemical bond, generally with concomitant hydrolysis of a small chemical group on one of the bulky molecules, or simply linking of two compounds together. Example, that enzyme that catalyzes the joining of CO, CS and so on. The next question relates with the chapter Human Health and Diseases. Select the option including all sexually transmitted diseases. A. Gonorrhea, malaria, genital herpes. B. AIDS, malaria and filariasis. C. Cancer, AIDS and syphilis. And D. Gonorrhea, syphilis and genital herpes. And the correct answer is D. Friends, gonorrhea is a sexually transmitted disease caused by bacterium Neisseria gonorrhoeae. It tends to infect warm, moist areas of the body, including the urethra, eyes, throat, vagina, anus. The female reproductive tract it can infect, the fallopian tubes, the cervix, and the uterus. It can pass from person to person through unprotected sex, anal, vaginal, or oral sex, and people with numerous sexual partners or those who don't use a condom are at greatest risk of infection. Malaria is a disease caused by the parasite Plasmodium transmitted to people through the bites of infected female Anopheles mosquito. What I mean to say is it's not a sexually transmitted disease. Genital herpes is certainly a sexually transmitted disease that causes herpetic sores, which are painful blisters, fluid-filled bumps that can break open and ooze the fluid. It is caused by virus HSV1, which causes cold sores, and HSV2, which causes genital herpes. Now, syphilis is a highly contagious disease that's mostly spread through sexual activity, including oral and anal sex, and the infected person often doesn't know that they are already suffering from the disease and may unknowingly pass it on to their sexual partner. Syphilis is caused by bacterium Treponema pallidum. The patient gets it through direct contact with the syphilis sore on somebody else's body, and this usually happens during sexual activity, but the bacteria can also get into the body through cuts on the skin or through mucous membrane. Now the next question relates with the chapter Evolution of Life, and the question is, embryological support for evolution was disapproved by A. Alfred Wallace, B. Charles Darwin, C. O'Parron, and D. Carl Ernst von Baer. And the correct answer is D. Friends, Carl Ernst von Baer's laws of embryology, or the laws of development, include 4 rules that were proposed by him to explain the observed pattern of embryonic development in different species, which disapproves the embryological support for evolution. The next question relates with the chapter Morphology of Flowering Plants, and the question is, the roots that originate from the base of the stem are dash. A. Primary roots, B. Prop roots, C. Lateral roots, and D. Fibrous roots. And the correct answer is fibrous roots. Look on the screen to understand the different types of roots. Friends, the primary root is an organ which forms at the root pole of the plant embryo. The root and the shoot that develop directly from a plant embryo are considered to be primary. Thus, in a young seedling, the primary root is that which continues the growth of the embryonic radicle. Now, prop roots are one of the modified roots for support, which develop from the branches of the tree, and they hang downwards and penetrate into the ground, thereby supporting the tree. Example, the roots of the banyan tree or the rubber plant. Now, what are lateral roots? These emerge from the pericycle, that is the meristematic tissue, and extend horizontally from the primary root, that is radicle, and over the time make up the iconic branching pattern of the root systems, contributing to anchoring the plant securely into the soil, increasing the water uptake and facilitate the extraction of nutrients required for the growth and development of the plant. The fibrous root system is the opposite of taproot system, usually formed by thin, moderately branching roots growing from the stem. A fibrous root system is universal in monocotyledonous plants and ferns. The fibrous root system looks like a mat made out of roots when the tree has reached full maturity. The next question relates with the chapter Genetic Engineering and Genomics, and the question is, in gel electrophoresis, separated DNA fragments can be visualized with the help of dash, and the options are A. Ethidium bromide in UV radiation, B. Acetocarmine in UV radiation, C. Ethidium bromide in infrared radiation, and D. Acetocarmine in bright blue light. And the correct option is A, that is ethidium bromide in UV radiation. Look on the screen to understand how it looks like. A Garros gel electrophoresis is used to segregate DNA fragments according to the mass and the size. Now, ethidium bromide is a fluorescent stain used in this technique. Ethidium bromide, when exposed to ultraviolet light, produces a fluorescent effect. Hence, the DNA tagged by it can be traced quickly on the transparent gel. The picture can easily tell you this. Friends, like if you have really liked the video, subscribe if you haven't, and hit the bell icon. Thank you so much.